Welcome to Mr. Chalk's Revision Tips. In this video, we will look at types of drugs, drug development and Flumidamide. So, new drugs are being developed all the time. Historically, drugs have come from nature as parts of plants and microorganisms have been extracted. So, certain drugs can be extracted from natural sources and have been used for a long period of time. So, for instance, willow bark was used by the ancient Greeks to help cure fevers and pains. It was later discovered that the active ingredient in it was salicylic acid, and salicylic acid is what is used to make aspirin today. So plants are still important today, but most plant drugs are now created in the laboratory at pharmaceutical companies. So three different variants of drugs. You can have stimulants. So stimulants are drugs that stimulate the brain and the nervous system, speeding up communication between them. Depressants slow down the activity of the brain and the nervous system. And hallucinogens interfere with the brain and the central nervous system in a way that results in something that distorts the perception of reality by the person who is taking them. So if we look at this question, so legal non-prescription drugs have greater impact on health of the population than illegal drugs. So it's just two reasons why. So things like more people take or use legal drugs are more readily available. Things like alcohol causes more liver or brain damage. Tobacco, which is available, causes cancer. And there are a range of other lethal non-prescription drugs that people might end up taking. So new drugs need to be tested and trialed before doctors prescribe them to patients. So three things that have to be considered. So safety. So it's important that drugs aren't toxic or that they don't have side effects that might be harmful to people. They've got to be effective. So this is known as efficiency, and checks now go and see whether it has more of an effect on a patient's symptoms than something that is already available. And the dosage needs to be looked at. So this varies and has to be closely controlled, as too high concentration of a dosage may be toxic. The drugs are tested using computer models and skin cells grown using human stem cells in a laboratory first. So this allows the efficiency and possible side effects to be tested. So many substances fail at the first test of the preclinical drug trial because they damage the cells or do not seem to work. Drugs that pass this first stage are then tested on animal in the second part of a preclinical drug trial. So in the UK, new medicines have to undergo these tests, but it is illegal to test cosmetics and tobacco products on animals. So a typical test involves giving a known amount of the substance to an animal, then monitoring them carefully for side effects. Drugs that pass animal tests are then used in human clinical trials. They're tested on healthy volunteers to check that they're safe. The substances are then tested on people with the illness to ensure that they work. And finally, low dosages of the drugs are used initially, and this is the safe dosage that increases until an optimal dosage is identified. So drugs must be tried before the drugs can be used on patients. So if we're looking at three reasons why, some of the reasons you could give would be to look for toxicity, to look for side effects, to see whether they interact with any other drugs, to see how well they work, and to measure the amount or the dosage needed. In the late 1950s and 1960s, pregnant women across the globe were given Flumidamide to combat morning sickness. So people had no idea how this chemical would act on early developing fetuses, so growing babies, because they did not test the drug on pregnant animals. So around 10,000 babies were born with disabilities as a result of their mothers taking the Flumidamide drug. So they all suffered from shortened limbs. So the drug was not only administered as treatment for morning sickness, it was also marketed as a harmless sedative.
Thanks for watching.